Welcome everyone to this Cleveland Water Alliance webinar in partnership with EQO. And today we're talking about genetic surveillance and early stage detection of harmful algal blooms. And we're going to be talking about using environmental RNA technologies and advanced molecular biology sampling. So try to say that 5 times fast. It's going to be a great conversation. We have a great panel. Thank you everyone for joining in. Just a few notes here when we get started. I want to introduce our panel first and foremost. So we have John Higley from the EQO, and he's the CEO, and he has 20 years of innovation in biotech, previous startup, exited by IPO. And I'll give John a better introduction when I hand it over to him in just a moment. We have Erica Schwartz Taylor, PhD, and she's an EQO co-founder. She's a senior science advisor, a molecular ecology expert. She's very excited about molecular ecology, so if you have any questions, drop them in to the Q&A. And she developed probes used as the standard by NOAA for toxic algae detection. And then we have Gary Anderson, PhD. He's a co-founder of Veriset, and he's a microbiology professor with UC Berkeley, and he has over 20 years of microbiome experience. And also on this call is Marie-Tay Ball. She is the EQO business development expert here. So she's going to be popping onto the chat and the Q&A. So if you have any questions as our panelists are beginning their discussion, go ahead and drop them into the Q&A or into the chat. So that Q&A function is just right down here. So feel free to drop in questions. We'll have about a half-hour presentation from these panelists, and then we'll open it up for Q&A. I know that everyone's going to have a lot of questions because this is a fascinating topic. So Marie-Tay will be in the chat answering your questions in real time, and we'll get to those after the discussion as well. And I'd like to launch a poll as we get started here. So feel free to answer those questions as they come up for you. We want to know where you're tuning in from, what your profession is, and are you interested in tracking toxin levels, what your interest level is there. So go ahead and fill out that poll as we get started. And now I'd like to turn it over to John. And just give me 1 second here.
Oh boy, that's happening quick.
It's happening quick.
Here we go.
Here we go. So John founded EQO with the goal of bringing the modern molecular biology lab to the field of environmental biology. So prior to forming EQO, John worked on early cancer diagnostic platforms and microbiome analytics at Thermo Fisher Scientific, and led the experimental therapeutics department as the senior director of R&D at Molecular Templates. John is a science advisor to the Interagency Working Group on eDNA, which includes representatives from the U.S. Geological Survey, Bureau of Reclamation, U.S. Fish and Wildlife Service, and multiple state departments of natural resources. So just a little bit of an expert here. So go ahead, John. I know you have your presentation. Feel free to share your screen, and we'll go ahead and get started. So like I said, we have that poll. Feel free to fill it out. Feel free to drop any questions in the Q&A or the chat, and we'll kick it off here. Thanks so much, John.
Okay, wonderful. Thanks, Sam. Much appreciated. Yeah, so I'm going to kick us off, and mostly I'm just going to kind of introduce to the high-level stuff, and then I'm going to let my my friends here, Erica and Gary, really get into the details. So I'll give kind of the overarching stuff. So essentially, what we're kind of working on with this project and this idea is around harmful algal blooms, and the history on this has largely been around the way we detect harmful algal blooms is by eye, by toxin after the fact, and by nitrogen input and things like that. These are all somewhat indirect, aside from, the eye is not really super helpful, and they're either very late stage or they're not detecting the actual issue itself. Coloration changes, things like that. Blooms are often very unpredictable, not just in their the quantity of cyanobacteria that blooms in a system, but also in the severity of it. So what toxins are being produced? How much problem is this really for the community and for the environment? And the what we really want to get at is there's a lot of variance in this system. There are so many different factors going into what is a toxic algae bloom and how it occurs. So our goal is to kind of detect this earlier and using biological metrics. So I won't go through this. We just went through that. Here's my team. They're great. Love them. Moving on. So Eco as a company, we have these automated sampling systems and these and some systems for field management. And Veracet, essentially the flow of this is that EQO, we have systems to collect biological information from the environment and can preserve it effectively. And then we have a laboratory where we can extract RNA and DNA very effectively without degradation and with removal of inhibitors quite easily. And then we can utilize our backup, our lab here at EQO and Gary and Veracet's lab out in Berkeley to do some more in-depth analysis of what's really going on and what toxins are being produced and what individual species are present in any given water sample. So at EQO, we're doing the front half of this, right? So we do sample concentration, collection, and preservation. We have a proprietary sample collection system and a proprietary sample preservation system, as well as additional proprietary stuff on the extraction and side of things. Essentially, what we're doing is we are concentrating a large volume of water over a series of filters in our sample cartridges in either an automated unit or a handheld unit. And then we're preserving the RNA and the DNA using a buffer of our own design that does not degrade in the environment given changes in temperature, pressure, or water composition and whatnot. So it's thermostable, preserves RNA for up to 6 weeks. In temperatures ranging from freezing to over 100 degrees Fahrenheit. And then we can extract that information or that we can extract that the nucleic acids on the back end on our side. And we have a extraction process that uses a flocculant to remove, hold on, the polls thing just popped up and it's getting in the way of my slide. That can remove inhibitors on the front end, and then we have a system on the back end to make sure that any inhibitors that are remaining don't interfere. So once we extract the RNA and we look at toxins, so we're looking at toxin expression levels, which Erica will talk about a little bit later. We also send the RNA and the DNA off to Veracet to do the second half, which is this side of things. So we're going to do the second strand synthesis, but we'll send it off to Gary to do the back end stuff. And the really cool thing on the microarray side, microarrays have been around for quite a while, but they weren't to the point to where they needed to be. And Gary's team has really gotten them to a point to where they're an actual diagnostic. It's a closed system. It works much more. It works much better as a diagnostic than a next-gen sequencing platform because it's reproducible and because it's a closed system. So he'll talk more about that side of things. The goal here is so essentially, how do we do this? This is kind of our automated sampling section. You'll get these slides. You can kind of look through this and see how this works and whatnot. But essentially, it's an automated sampling platform that does the things that I told you. This one has 12 sample cartridges on board that you mail back and forth to our lab. We also have handheld sampling systems. This is kind of the early prototype version. The new one looks way cooler than this. We have one at Limnotech right now, and we're going to be doing the Cleveland Water Alliance test beds to do some work on the handheld samplers. And this is for like geospatial. So on the automated sampler, it's kind of a semi-permanent installation. It's for a place that you know is going to have issues, and you want to sample on a regular basis for like a temporal space to figure out eDNA and eRNA, all kinds of things. It doesn't matter what you're sampling for, but all kinds of invasive species, native or biosecurity threats. This one is for you want to get on a boat and take a bunch of points around a lake and understand a baseline at a single point in time. So we have geospatial and temporal spatial applications here. The process is, it gets collected, it gets preserved in one of our sample cartridges, which is what you see in the second step here, and then we analyze it in the lab. So the important thing to recognize here is that this is not all getting analyzed on the machine, on the instrument that's doing the collection. This is very sensitive work. RNA degrades very quickly. The reason why we're going to be talking about RNA a lot today is because eDNA you may have heard of, but DNA is a very stable molecule. It doesn't break down very easily or very quickly. Um, but DNA is kind of like the book of all blueprints that you could send to a contractor. The RNA is the message that you're actually telling the contractor what to build, and then the protein is the thing that gets built, right? So what we're doing is we're intercepting that message, and it degrades within 40 minutes of being of occurring. And so we collect that, preserve it, and then we can take kind of an environmental snapshot of what genes are being expressed at the time. That allows us to understand what toxins are being expressed, what's alive, are there signals for breeding, all kinds of things that you can query the transcriptome, as we call it, to figure out what's going on at the time. And it has to be utilized in a lab to be done effectively or with any level of cost, because the robotics involved in doing this in the environment are quite beyond us as far as being financially viable. So we have to utilize it in the lab and have experts work on the RNA, just because it's very difficult to work with. It's a finicky molecule. You look at it funny, it falls apart. So the goal here is forecasting. The idea being that if you catch, just like with cancer, if you catch it early, like prevention is cure, early diagnostic is cure. This is the idea with in oncology that we've been working on for a very, very long time. And right now, we don't really notice an event until it looks something like this. And at that point, yeah, you can analyze how much toxin is there, and you can analyze how horrible it is. But we could have prevented this from happening if we utilize some earlier detection techniques. So what we want to do, instead of analyzing this whenever the cyanobacteria is already out of control and when the toxin levels are already out of control, we want to identify the toxin levels as they increase. So when they first spike up, when you never see, again, think to cancer, before you could palpate a tumor, when do they first show up? That's what we're going for here. And there are some things on the market now, and there's new stuff coming out. AECOM just released a really cool article about a new project they're doing to remove toxicology. And if you catch it early enough, you can actually prevent these kind of things from causing public health concerns, water quality degradation, and all of that. So we're just going to move the forecast model from when it's a true problem to when it's an actual bloom to when we start to the point at which, or right before the point at which the bloom begins. That's what we're trying to do, is just kind of move that window. And that way we can forecast much earlier. And the way that we're going to be doing that is looking for toxins and for looking for changes in the microbiome. So now I'll hand it over to Erica, and please let me know when to advance the slides, and I hope I wasn't too long.
All right, cool deal. I hope you can all hear me okay. Thank you, John. Yeah, so I'm here to briefly talk to you about some ecology. John talked about forecasting and how we want to advance our forecasting capabilities by using some of this RNA technology. Well, forecasting is really complicated, right? And the reason why forecasting is really complicated is because cyanohab ecology is really complicated. As you all know, HABs form due to a variety of things, which is really what this diagram here is showing. This is not to go over every aspect of what you see in this diagram. It's simply to show that there's a lot of things that happen that lead to a cyanohab, specifically things like nutrient inputs, right? Temperature, light availability, even grazing. For example, with zebra mussels being in an aquatic system, those systems tend to be more prone to cyanohabs, and that's because they filter feed all of the cyanobacteria's competition and predators. So cyanobacteria can just have a free-for-all. But anyway, so this is a very complicated thing. So what we can really do is we can use RNA technology to at least focus on the cyanobacteria of interest itself earlier rather than later, to look for signatures in the water column that occur prior to a bloom. And I'll leave it at that. I could talk about ecology all day. But John, go ahead and advance to the next slide for me so I stop talking about it. Thanks. Okay. This slide is really just to put in an organized fashion the toxins that we know are prevalent in cyanoHABs. This is not a comprehensive list of toxins. It's also not necessarily a comprehensive list of genera, which you see on the right-hand side. The genera represented here are just the ones that we think of most probably when we think about the toxin on the left-hand side. And again, there's a couple of other toxins too that we're not really getting into here. But you can see that we have a mix of hepatotoxins and neurotoxins that we deal with, which of course, you know, as you know, lead to huge public health concerns. So let's talk a little bit about what we know about these toxins. You know, if I had all day to talk to you, we could go through each and every one of these in the studies that have been done. But for the sake of time, I'm really just going to focus on microcystin because that's really seems to be the most well researched as far as cyanoHABs are concerned. And a lot of studies having to do with gene expression, nutrients, light, and those types of things. All right, next slide, please. All right. So again, we won't get too far into the weeds and all of this information. There have been a lot of studies. There are some contradictory studies out there as well, as far as the current, or as far as what's known about environmental effects on toxin production anyway. So environmental effects range anywhere, you know, from anything like pH, temperature, and as you see here, nutrients and light. But here we're just focusing on nutrients and light because there's a pretty good wealth of knowledge behind these two environmental aspects. So nutrients, and specifically nitrogen, seems to be really important in microcystin concentration. This makes sense because the structural molecule of microcystin actually requires like 10 atoms of nitrogen per molecule. So, you know, you need the ingredients to build the molecule. So the link there makes sense. However, there have also been gene expression studies that show that the genes that code for the various components of microcystin are upregulated or downregulated depending on nitrogen concentration. So there's a study that showed, you know, in nitrogen deplete conditions, gene expression of those genes, the MCY genes, are downregulated by a significant amount compared to normal nutrient conditions. Similarly, high light conditions is where we typically see a positive correlation between microcystin production and also growth rate. So this kind of lumps in number 3 on this slide as well. You kind of inherently talk about all these together. But in highlight conditions, we find that not only do photosynthetic rates increase, but microcystin concentration also increases in the cells. And they think that microcystins likely have some oxidative stress protective mechanism. So that's why we see them at these points where light is high, growth is high, photosynthesis is high. And so microcystins are kind of conferring this protective benefit. You know, we can really pull all of this together, the nutrients, the light, and the cell growth with toxin production, because in practice, you know, microcystin concentration is typically the highest during the earliest or during the earlier phases of a bloom, where there's nitrogen replete phases, and also where the photosynthetic rates are high. And so this really supports kind of the fact that all of these likely play a factor in microcystin concentration. You know, and there have been similar studies performed on other toxins like anatoxins, saxitoxins, nodularin, and those things. But again, you know, we don't have time to run through each and every one, but there's some very interesting things out there. And I'm always happy to share resources as needed. So the other thing that we've seen in the literature a little bit is that sometimes toxin gene expression and toxin concentration, there seems to be a disconnect between the two, which might be somewhat counterintuitive. There does seem to be a pretty strong correlation between gene copy number and toxin production. However, you know, we want to use RNA technology because we want to see what's happening now. You know, it's present in the environment now. Because with DNA, it's got a longer resident time. But some of that disconnect between gene expression and toxin concentration can actually be attributed to the fact that the toxins tend to bind to protein. And when that happens, they likely are not being detected by the same methods that are able to detect the toxins that are not bound by proteins. But there is definitely still a little bit of a gap in knowledge there and how directly you can measure gene expression and toxin concentration. It looks really promising. There's just some kinks to kind of work out in the system there. Okay, next slide, please. Okay, so just to give an example, we can take advantage of these genes that code for the toxins. Here is just two examples. This paper that's cited here actually has a diagram for each of the different toxins if you're interested, so you can see the operons and the cassettes from both. But we can take advantage of the conserved nature of some of these genes within the cassette. So what we have is we actually have these MCY, I was going to say, so used to talking about myco things. Anyway, MCY, so the microcystin gene cluster. This spans about 55 kb in the genome, and it consists of 10 transcribed open reading frames and 2 major operons. The first operon being A through C, which are these blue guys right here, and the second operon being D through J, which are these guys over here. What we can do is we can take advantage of the fact that non-toxic cyanobacteria actually won't have these MCY genes, even though that's not totally true. Some of them are lacking, some of them have partial copies of some, it just varies by species. But so we can actually create a probe using something like the MCYE gene. So we can actually detect if that gene is being actively transcribed or not in the environment. And we could do something similar with something like nodularin, right? And by using the NDA gene of its cassette. There's one other thing I was going to say and I totally forgot. Oh, also, the other thing that's nice is that we know that all of these things are going to have the 16S ribosomal RNA or ribosomal DNA, depending on how what terminology you like to use. So we can actually use a more generic marker here and then add in more specific markers to focus on those species that produce the toxins. And I think that's probably it for my time. I don't want to cut into Gary's time too much. So these are just a couple of examples, but here are some of the genes that we would use. to focus on some of the other toxins as well. Okay, all right, Gary, take it away.
Oh, looks like you're
I
muted.
guess I'm muted. Sorry. Yeah. Thanks, Erica. And so if I could advance to the next slide, please. So yeah, as John introduced before, Veriset, of which I'm a co-founder, is a water microbiome company. And what we do is we provide the actionable data to our customers. And we do this so that they can actually solve their problems. And how we do it is we use proprietary DNA and RNA technology to identify all the microbes, as well as unique analytical methods. And we can do this to assess risk of contamination. So next slide. So we have, our technology was developed in my lab here at University of California at Berkeley. And it uses a microarray based approach to measure all microbes, both by using DNA and RNA. And so on the upper left corner here, you see an example of an earlier version of our, what we call a phylochip, our microarray method. And so on our latest versions, we have up to 1 million different probes to detect any of the tens of thousands of bacterial species that could be present in a water sample. And so this is the only method that is able to actually use statistics to confidently identify specific types of contaminants. And we have experience then over the years of using this then to distill through the many millions of bytes of information that we get. So we have a fire hose of information, and we distill this down to a concise report that identifies what the problem is. We're internationally recognized as leaders in this area, both in my lab and now through this company, Veriset. About 10 years ago, we started in the water area using microbiome analysis with the BP Gulf of Mexico oil spill, where we were, my lab was the first to identify, using this microarray technology, we were able to identify all the species that were degrading the oil in the deep ocean. And in addition, we were the first to actually, we were able to identify specific contaminants at very sensitive levels based on the microbiology composition. So we then took this through Veriset to be able to identify at first fecal contamination in water. And we have used this with many customers. For instance, the EPA, the US EPA, as well as regional EPA labs have worked with us. In addition to water boards, we have successfully solved their questions in identifying sources of contamination, especially fecal contamination. And we just finished a project with the Department of Health in Hawaii, where we solved a problem that had been ongoing for 10 years of controversy about who was creating the contamination. So next slide. So as both Erica and John have mentioned, harmful algal blooms are increasing in their problems. So why is this? So we actually know how to mitigate these algal blooms, especially in fresh water, by just reducing the nutrient sources that help to cause them. We have treatment methods that are specific to treat these cyanobacteria. But the weak link is the upper link here, the monitoring. So most monitoring methods that are currently being used are more indirect. So we can do satellite monitoring. We can indirectly measure chlorophyll content or look at the nutrient levels. But what's really lacking is a more detailed, in-depth look at the microbes actually involved in this process, especially the cyanobacteria, the freshwater harmful and non-harmful algal species that are present. And so, next slide. Yes. So we do this by looking at the water microbiome. And so I wanted to just first go over what is a water microbiome. So most of you have heard of the human microbiome for a number of years now, and how that really is important for our health. So in the similar way, water microbiome is a large and diverse collection of microbes specific to a source. In this crude diagram here, it's a lake source. And there are tens of thousands of different species here. I just color coded them based on the different types. So for instance, having microbes in your water, especially bacteria, it's not a bad thing. It's actually, they're always present. And so a lot of microbes are associated with healthy, pristine lakes and other water systems, and we can identify those. There are many different types as represented by the different shapes here. In addition, we can have cyanobacteria causing algal blooms, and so there are many different types of those organisms which can be present. In the purple shapes here indicate some of the types of microbes that are specific to the particular lake chemistry. So for instance, if you have more or less phosphorus or nitrogen in your lake, you're going to have differences in both types and abundance of specific organisms. And in addition, I just put a few symbols here to represent specific types of nutrient pollution coming from, say, a horse or septic system, and even we can identify the specific pathogens. So altogether, this entire grouping of organisms is called the water microbiome. Next slide. And so that's what we do. We identify all of these organisms, and we develop what we call a microbial community fingerprint. So this is just one of the ways that we look at the sample here. So on the x-axis here, each one of these horizontal lines, vertical lines, represents an individual different species of bacteria. And I've color-coded them based on their different, just broad general types. And the height of the bar represents just the abundance, or in this case, the fold change, the increase in abundance over a baseline condition. So in this actual sample that I have here from a real contaminated sample, we were able to identify specific microbes that were present and were diagnostic to this particular contaminant. So next slide. So in the same way, we can do this with cyanobacteria. We can track over 150 different species of cyanobacteria simultaneously at the same time that we're detecting the tens of thousands of other bacteria that are present in a sample. And so a similar type of fingerprint is shown here. In this case, this is just what we would expect would happen. So this is more of an idealized instead of an actual sample here, in which we're looking at about 150 different cyanobacterial species, and the y-axis in this case is the log of the abundance of that exact species. And so again, just at a fingerprint in time, we can identify which of the species are present and how abundant they are. And from doing that, we can actually start to look at predictive tools for identifying when these cyanobacteria blooms are about to happen. So as both John and Erica mentioned with RNA, that provides us with an exceptional tool here that really has not been available before. So some of these techniques have been available just for a few years because the cost is finally down to where they're affordable to use in environmental situations. But the using of RNA really hasn't been done until now because it's just been too difficult to get RNA from these water samples. But now with EQO has solved that problem. We can actually identify all the different types of organisms, not just by the DNA, which identifies that they're present, but actually by their activity, which gives us a whole new level of real-time identifying what's going on in the system. And so that's really what we aim to do here, is that in the end, this can be a very interesting scientific project, but what our ultimate goal here is to be able to develop a very sensitive diagnostic tool that is able to go in and predict these algal blooms before they happen. So with that, I'll give it back to John.
Sorry, I was muted. Thank you, Gary. I did not mean to. I accidentally advanced a slide. That was not a curtain call. Anyway, thank you everyone for attending. We're really excited for the Q&A, and thank you for the Cleveland Water Alliance for having us. To kind of wrap up, the idea here is that we can detect a whole host of species and toxin expression levels, and that's really what we should be detecting for forecasting models. In addition to looking at those things, we'll also be analyzing all the traditional information that's collected by the people who are already taking in the water, the water treatment facilities. So we'll also be analyzing nitrogen levels and temperature and turbidity and all of the dissolved oxygen content. And the point here is that to take all of this information and put it through kind of an AI/ML system to create a proper forecasting model that can be really effective at preventing public health outbreaks. So with that, I'm sorry for being so long-winded, and now we will take lovely questions. Also, if anybody wants to contact us, I would very much appreciate it. Contact Marite. She's the best out of all of us at keeping up with everyone. And here's her contact information. I'll leave that on screen for the Q&A. Thank you so much.
All right, thanks everyone. Got some questions coming in. Everyone feel free to use the Q&A or the chat function to submit any questions. And no question here is dumb. I know that everyone is tuning in with different levels of experience from different lenses. So please feel free to ask any question. If something isn't making sense or if you need clarification, please feel free to ask, because if you're thinking it, someone else probably is as well. So I'd like to pose the first question to Gary and maybe Erica. So why is it seems like we're hearing about harmful algal blooms in the news more and more? We have the red tide in Florida, and we're hearing about these large scale issues. Why is it happening more frequently, or are we just hearing about it more?
Yeah, I'll start, and then Gary, you can add anything that I might leave out. So I would say that they are happening more frequently because, things like weather patterns, right, make a difference with harmful algal blooms. Things like climate makes a difference. So with warmer temperatures, we get certain things, in this case, cyanobacteria, that outcompete their eukaryotic phytoplankton counterparts. And they really thrive in just, hot conditions. With things like, zebra mussels in a lake system, for example, like I mentioned, that can also add another layer of complexity, I guess, when it comes to why we would see blooms more frequently. Just having those there in large numbers certainly do that. More rainfall, more runoff, more hurricane activity, more nutrient mixing, those types of things. Gary, do you have anything to add?
Yeah, I think it's really interesting because we used to, like, for instance, Great Lakes, Lake Erie, for instance, which a lot of you are interested in, used to have a lot of harmful algal blooms up until the 70s. And this was because they were directly dumping sewage into the lake, and that went away. And so it was amazing. It was really a success story. The algal blooms went down and down and down. And then about 10 years ago, they started, or around then, started coming up much more frequently. And so why is that? So we have no more direct sewage going in, but we have unidentified sources of contamination going into these water bodies. That's probably the main cause, exacerbated by the climate change. That is why that, although we've gone down from the 70s through the 2010, we're now every year going up. Yeah, that's probably why.
And I'll add just a little bit to that, is the fact that also the types of algae that are blooming now versus what was blooming between the 1950s and the 70s are different. So back then, it was nitrogen fixing bacteria that would bloom, and now it's non-nitrogen fixing bacteria that are blooming, suggesting that they're getting nitrogen inputs from somewhere because they can't make it themselves. So it's interesting, as Gary said, to see that dynamic of how things shift, right? Given different environmental inputs and time, things of that nature.
Yeah, we had a follow-up question. Is sediment runoff, does that contribute to harmful algal?
Absolutely. Yeah, it's especially, as many of you know about the reactive phosphorus that is causing problems. So different agricultural conditions, again, we know how to mitigate these types of blooms. It's just we're not doing a good job in monitoring to prevent, once we see something happening, to really go in and try to solve the problem before it gets worse.
Another question that came in, upon sample collection, how long does it take for the sample to be analyzed in the lab, and how much is the cost involved for the analysis?
Sounds like a me question. So two things here. One, it takes the shipment to us, to our lab, and there's a little bit of time there, usually like a day or two, just for FedEx to do their job. And then our extraction process and analysis takes 3 days max. And as soon as we do the extraction, we send it over to Gary. There's a little bit of a shipment there as well. That's an overnight because we have to keep it cold. So then there's a day involved there, and then Gary has his analysis. And the analysis on that end, the full microbiome thing, is something that we're still working to compress. I can't give you a hard number on how long that side of the information takes, but all of the other stuff is pretty quick, within a week. And on the cost measure side of things, we do well. So right now, the purpose of this, and kind of what we're talking about right now, is this is something that we're actively pitching to the Great Lakes Protection Fund. And the size of that project is it's substantial, but it's not too high. On a individual auto sampler basis, the way we sell this is not per sample; it's as a subscription service. So the cost for that, we can talk to Marite about kind of what the quote is. But typically, you're paying a fixed price. It's under 6 figures per year, and it's it's up to 100 reads or 100 samples per year that can be analyzed using that. So that's kind of the cost structure and the turnaround time.
And so then, John, the test, it's not one test per individual species. It's one test that covers all of the species.
Well, so what's beautiful about this is we can always go back and recheck. So what we're doing on our end is we're looking for specific toxin extraction.
Getting a little bit of feedback, or we're having a little bit of trouble hearing you right now, John.
Oh, I'm so sorry. Can you hear me okay now?
It's still
okay?
Just a little bit.
Not great. No, I apologize. I don't know what I can do about it in my setting. But what I was saying is, what we're looking at is individual toxin expression. What Gary's looking at is every species in that system. And the beauty of this process, though, is that once you take the sample, once you have it extracted, you can reprobe for anything of interest. So from the same sample, you're like, hey, I wonder what my agent carb situation is. We can reprobe for that. It's not a problem. It's just a different probe.
Great, thank you. I can hear you a little bit better right now. Another follow-up question there. Okay, a few people are still having a little bit of trouble hearing you. So a follow-up question, to that, in your experience, is there a specific period between early detection and use of one of the emerging treatment methods to successfully prevent or minimize a HAB?
This is something that we don't fully know yet. This is something that the kind of the point of this is to learn how early we can detect. What we do know is that there's good evidence to support the concept that early detection is prevention is cure. We know this in cancer studies. We know this in invasive species. We know this in HABs from previous information. The specific timeline, I can't give a number for that, but this is something that we can learn with additional work.
Here. So, as a practical matter, what new management action will better forecasts of HABs enable?
I will, I'm going to defer my colleagues here in a second, but I will give a quick shout out to AECOM. Had a really cool announcement about a week ago about a new project that they're doing to remove harmful algal bloom materials. There's also LG Sonic is in the game. There's some specific algicides. There are a variety of treatments. And with that, I'll hand it off to Erica and Gary if there's anything you want to add. What I do know is that in general, the sooner you treat a problem, the better chance you have of being successful.
Yeah, I could just add to that, if you if you wait too long, you cannot use any algicide or anything to treat the algal blooms, and that's really our goal here, is to be able to detect it at an early stage where very minimal chemicals could be used. So you're not really doing minimal environmental damage, and at the same time, if you when you kill the cyanobacteria that are producing these toxins, they release a lot more toxins into the system. So if you're doing it at bloom time, it's just too late. You're gonna make a toxic soup by doing that. So that's really that's the overall. That's what's so important here, is that there really haven't not been good methods to do this early prediction, and that's really where we think that based on our track record, that we can really make a difference.
Yeah, and that's to even build further on that. I mean, that's the point there that Gary was making about when you lyse these cells, they release toxin. When you break them open, the toxin gets released. This is what happened in Austin. We had that deep freeze that probably everybody out of Texas heard about, and that did was it lysed a bunch of cyanotoxin that was the filamentous type that produces anatoxin-a, which is a real problematic toxin, and it just lysed a bunch of cells, which caused a toxic event. And this is the kind of issue, is that if you attack a bloom too late, you're just releasing their toxins into their environment. And so you really have to hit it early so that the toxin doesn't actually cause any problems. So old Paracelsus quote, right? Everything is a toxin given the dose. And if you can interrupt these organisms when there's less of them, you have less toxin, and it is not toxic.
So following up on that, and Erica alluded to this earlier with the different kinds of blooms that have happened over the years. Not all blooms are toxic. Are you concerned with monitoring non-toxic blooms? Do you come across non-toxic blooms very often?
Are we concerned? They happen, and that's the kind of the whole point. It says, like, again, going to the Lake Erie example, sometimes they have very large blooms and they produce very low levels of toxin. Other times they produce very high levels, and that's really the missing piece here, that what Erica was saying, the ecology, that there's been extremely few studies on. And so we believe that there's a link between the types of microbes that are present, the types of cyanobacteria that are present, and the genes that are present, that all combine to either make for a lot of toxin being produced or a little. So it's just, it's not just the species that are present, because the species can be present and still the toxin isn't produced. So it's a much more complex matter than has originally been attributed to it.
Yeah, this is a multivariant system, so we have to kind of tease out the variants to figure out exactly what's going on.
Mm-hmm. Yeah, a few more questions in here. Question for Gary. Does the microarray of HABs taxa target various toxin-producing genes, or does it target a typical barcoding gene, such as 16S?
Yes, it's the barcoding gene. So we're detecting the cyanobacteria in the rough manner. So that's why I was saying we're detecting around 150 different species. So we're not getting down into the substrain level of detection just in our overall look at the microbiome. And so that's where the EQO and the genetics of the specific systems comes into play. So it's very important to know what are the types of cyanobacteria and how are they coordinating with each other. Are some going up and some going down? That gives you clues to a bloom happening or not happening. And then within there, is it going to be harmful? And so knowing some of the genetic information that's going on, and especially with RNA, again, I just can't emphasize that enough. That's a real game changer here. To be able to do RNA, it just has not been done before. And that's for the first time looking at activity. So that's how we can really find out if it's harmful or not.
And another one here that I'm going to read verbatim because this is beyond my my language speaking skills. I feel like I'm speaking a foreign language right now. Are there any issues with matrix? For instance, high DOC T-stained waters or high suspended sediment? And you can read that one in the Q&A, Gary, too, if that doesn't make sense the way that I read it.
Yes, I'll take that. T-stained waters are suspended. Well, from the collection side of things, there is an issue to an extent with collection of things that are turbid water or water where there's a lot of biological activity, just because the sample cartridges can get clogged. So with the automated sampler, the way that it works is that it senses the outflow pressure. And so when that drops below a certain point, it will do a backwash, just kind of mix things up a little bit so that it can have more stuff flow through. And if it does not reach its programmed number of liters that needs to flow through the sample cartridge, it'll switch over to a second sample cartridge, draw more water, and then send a signal to the person collecting it saying, okay, marry these two samples. I used to work I was a chef many years ago. We used to marry the ketchup, so that's not legal anymore, but that's what we used to do. This is the same idea. Marry these two samples. It's the same sample. It's just we can only get 250 liters over this one, so we got the other 250 liters over this one, or the first 750 and the second 750, whatever it is. And that's how we kind of deal with that. Now, as far as interference on the back end, no, we don't have an issue on that because we're removing inhibitors on the front end of the extraction process with a flocculant that removes 90%, if not more, of the inhibitors that are in the water sample itself. And essentially, we kind of run all the water through. The filters are somewhat semi-dry, and then we put in our preservation buffer. So they're basically suspended in the preservation buffer. And that's we don't care how T-stained it is at that point. We don't care what other inhibitors are in there at that point because we've stopped inhibition, we've stopped RNA degradation.
So then along those lines, if someone was interested in toxicity of soil, can you test just soil, or do you need the water?
We can test soil, yeah. It would be a different collection practice, and you'd use a different. The collection practice would be different, and we would have to validate that. But that's certainly within the frame of reference, and I have certainly utilized soil samples.
Great. I think we have time for just a couple more questions. We have another one here that I believe we alluded to previously, but I just want to make sure we address it. If it is possible in some way generalized, how soon will the toxin be detected by chemistry analysis once it is detected by gene expression?
I think that's a question that we don't quite have an answer for yet, unfortunately.
Yeah,
chemical analysis, typically, if you're really trying to do, the gold standard for this is mass spec, right? You're going to be running a mass spec to determine the exact type of toxin. But even with that, there are limitations. As we alluded to before, if it's bound to a protein, we're not going to detect it properly via mass spec. And so there is a disconnect between gene expression and protein content as analyzed via mass spec. So there's limitations to every form of detection. The point of this is the idea here is that you want to get as many viewpoints as possible. There's that old adage about, a bunch of blind people touching an elephant trying to determine what it is, right? This is every diagnostic has limitations, and you want to get as many visuals as possible, have those visuals talk to each other to figure out what the real picture is. And that's what we're working on.
If I could just add to that, John, too, so in our previous work on hydrocarbon detection, it's using DNA and RNA are often extremely sensitive because you can detect things that, just when they're beginning to be expressed. And so in this case, again, as John said, we don't know, but the methods we're using are able to detect at very sensitive levels of toxins, sometimes maybe even before other methods.
All right, I think that brings us to time today. I just put Marite's information in the chat, and she did as well. So let me I'm just gonna share my screen again really quick with just some final information here. And thank you so much for all of the great questions. Thank you to all of our panelists today and our partner at Cleveland Water Alliance, our partner EQO. We have really enjoyed working with EQO on a lot of different projects in a lot of different ways. And thank you so much, Gary, for bringing your expertise with Veriset here as well. We do have, CWA does have another webinar coming up next week about with our water data forum, our monthly series. This is about managing your pipes, data-driven distribution network optimization. So we will be sure to send the recording of this webinar out to everyone who has registered. Feel free to forward that to folks who could not attend. And I'll also send registration information for our next webinar, as well as Marite's information there as well. So thank you once again. Thank you, panelists. Thank you, everyone who tuned in today. And thanks for all the great questions. So thanks so much, everyone. Have a great day.
All right, thank you.
Thanks, everybody.
Thank you.
Cheers.